So we are going to continue on this set of notes. The last thing we were talking about is the fluid compartments of the body, right? Yes? Okay, we've got balls. ECF and ICF. And which one is a larger component of the total body water? ECF or ICF? ICF. ICF is about 2/3 of the volume, okay? And then ECF has two categories of ECF, right? There's two different kinds of ECF. What are those? What? Interstitial fluid and blood plasma. Okay, so you've got intracellular fluid, and then you've got ECF, which is composed of interstitial fluid and blood plasma. So my question to you is there exchange between these three things? Or are they completely compartmentalized? There is exchange, right? Otherwise you could, for instance, never get the oxygen from your respiratory system across your respiratory epithelium and ultimately all the way inside your little finger cell for cellular respiration, right? So there must be exchange, but there is separation. Okay, so with our body fluids, the next thing that we are going to talk about is what you were doing with your body fluids. So last thing we said last time was that the body spends the vast majority of its time regulating one of those two categories of fluids, ICF or ECF. Who remembers which one and can explain to me the logic? Go ahead.
Isn't it ECF? Because ECF is only like one to two big pools of water, right, and ICF is like trillions of little pools, and exactly, it starts in the ECF.
Yeah, it starts in the ECF. Those are both really good explanations as to why the ECF. So it's a big circulating pool, and if you detect it and correct it in one place, then basically the corrections will get filled everywhere. And then, of course, it's a better time to do it before you actually have caused a problem with functionality. So the body spends about 60% of the ATP that you actually expend just maintaining homeostasis, which is pretty encouraging because that means that, like, the cheeseburger that you eat, you don't have to burn the whole damn cheeseburger. You burn 60% of the cheeseburger just staying, like, structurally human, right? 40% of it is how much you have to burn. The problem is, of course, we generally don't stop at a single cheeseburger. We have some fries and a Coke with that, and we supersize the damn thing, right? So that's the problem, is when you take it way too much. But 60% of your energy is just maintaining homeostasis, because the default is that your body would just pretty much go to entropy, and everything would pretty much become the same as the external environment. So you spend a tremendous amount of energy maintaining homeostasis, and primarily what you're doing is trying to maintain the homeostasis of the extracellular fluid so that it will take care of the intracellular fluid, right? So what we're going to learn today is how. How do you maintain the homeostasis of the extracellular fluid? So if you didn't monitor the ECF and it got to the ICF and caused massive changes, then you would have changes in function, yeah? And then you get potential for pathophysiology. Does that make sense? Okay. So physiology. Still, just so you know, we don't understand all of physiology. What we have is a really strong framework for how it works and what the big ideas are, but we don't have every single one of the details, right? We're still learning more details. For instance, the most interesting part of physiology to me is neurophysiology, brain function. And it has been somewhat of a black box for us. We haven't been able to figure out the physiology of the brain for a very long time. And we are learning over this last 10, 15 years tremendous amounts about the brain physiology. And the reason I'm telling you that is because I'm telling you, when you get finished with physiology, you're not going to be done with physiology. You're going to have to continue educating yourself in order to be a good clinical professional, right? You don't just stop learning when you're done with the class, because then your knowledge is going to be 15 years old, 15 years from now. So I will try to teach you the best, most current information that I have, but there's going to be more. There's going to be more next year. And students find that incredibly frustrating. I find it incredibly interesting. I love it because learning is my favorite thing. But some students are like, oh my God, the book says something different. And I'm like, I'm really sorry when what is printed doesn't agree with reality. Sorry. Okay, So whenever you see this in your textbook, I'm sorry, in your notes, what it means is that if you are a visual or maybe a kinesthetic learner and you like to do things and see animations, interactive physiology, which I told you guys about last time, which is part of the online component, it comes with a new textbook, has an exercise that I looked through and thought was relevant and perhaps useful. Do I ever test off of those? Nope. I just looked all the way through it and found the ones that I thought were relevant to the stuff that I was talking about so that you could use it if you want to. But I won't test off of it or anything, it's just helpful sometimes. Okay, so now how do you maintain homeostasis? Well, the primary mechanism by which you maintain homeostasis is something called negative feedback. And negative feedback, what it does is when a variable, like let's say body temperature, starts to go outside of the normal range, negative feedback will oppose it and try to bring it back into normal range. So in order to understand negative feedback, what I need you to do is dump your definition of negative that you typically use, because the definition of negative that you typically use has something to do with not good, right? Bad, or less than, right? Lower or bad. But that's not what this means. Negative feedback means that, for instance, if my body temperature starts to go up, negative feedback will bring it back down. But if my body temperature starts to go down, negative feedback would bring it back up. So in this instance, negative feedback implies opposition or negation. You are going to do the opposite of whatever is trying to happen to the body, right? So it's not bad because it's homeostatic, and it's not always down. So let's look at what a graph of negative feedback might look like, and we'll talk about whether it returns it to a set point or a range. So when you look at a graph of negative feedback, first let's talk about physiological graphs. The most common kind of physiological graph that you'll run into is something that tracks a variable, doesn't matter which one. And this time, let's do body temperature, because you all have a lot of experience with that one, and it will sound very familiar. We always put time on the x-axis and whatever variable it is on the y-axis, and that matters. And the reason it matters is because the x-axis is the independent axis, and the y-axis is the dependent axis. And that means that this graph, the way that it's set up, says that body temperature varies over time, which is true, yes? And if I flipped it, it would say that time varies with my body temperature, which would be amazing, and maybe you want to write a sci-fi novel about it if I can change my body temperature and change time, but I haven't written it yet. So you have to get these right, okay? So let's look at what a graph of negative feedback would look like for body temperature over the course of maybe a 24-hour day, okay? And this is just an example. So let's say it's an ideal day, and you get everything done that you wanted to get done, so you can go to sleep at peace, and that happens so rarely. Can't even remember the last time it happened. So here's a really, really good day. Okay, you're going to get up whenever you damn well please, right? And then your body temperature is going to start going up, right? You're going to sit around on your computer and fart around for a few minutes because nobody's making you do anything. And then you're going to make and eat breakfast, yes? Okay. And maybe, this is a very good day, right? You're going to work out before work. Yeah, how often does that happen? And then you're going to get in your car, and your body temperature is going to drop because you have to commute all the way to work. And then you're going to get to work, and your boss is going to talk to you, and it's going to be really irritating. And then, let's say you're me. You're going to go teach, and it increases your body temperature. And then you're going to go back and sit in your office for a little while. Then you're going to sit in your car and go home. And then you're going to go, somebody's going to make you make dinner, right? And then you're going to eat your dinner. And then you're going to watch HBO for an hour. And then you're going to have sex. And then you go back to sleep. Okay? So this is a day in which you checked all the boxes. You got every single thing done. So what's going to happen is you're going to see that if you look at negative feedback over time, you can start to see the suggestion that you're trying to stay in a certain range, right? Whenever you get out of the range, you start to pull yourself back in either way, right? So if you take a whole bunch of data points over a long period of time, you can get the good suggestion of a range. And if you take 30 days worth of data points, you're going to get a really good idea of what my ideal range for my body temperature is. Just from this set of data, I would say that it's probably right about here. you see that I keep trying to bring it back in right about there, right? Does that make sense? Okay. So what's going to happen is you do go outside of your ideal range for body temperature. And that doesn't mean that you're diseased, does it? What would mean that I was diseased in this situation?
Stay out right now.
Stay out. Yes. You always go out. It's part of the thing. It's just that you bring yourself back in, right? And that is your homeostatic control mechanism for negative feedback. So negative feedback is going to, every single time you go out this way, it's going to bring you back in this way. But if you go out this way, it's going to bring you back in, right? So whatever it's doing, it's negating whatever change occurred to help maintain. Does it look like it's trying to maintain my body temperature at a set point, 98.6, or in a range? Yeah. So maintain my body temperature at a set point, which is what we have memorized, right? We have 98.6 memorized. It's easy to memorize one number. But that would be like trying to maintain your house at exactly 74. And that's not what your thermostat does. Because when your house temperature goes up to 74.2, do you turn on the air conditioner? No. When it goes down to 73.8, do you turn on the heater? No. You can see how incredibly expensive and inefficient that would be. So even though we set it at 74, obviously there is an acceptable range around 74 in which you're not going to try to change it. Does that make sense? And you know, because you pay your damn bills, how expensive that would be if that had to occur. So your body doesn't do that either. It doesn't start to sweat as soon as you vary in body temperature just slightly too high. It doesn't start to shiver as soon as you vary slightly too low, right? It starts to institute changes when you go out of the range, but the range, not the set point. Okay, so what we're going to talk about now is how do you do this? So what's going on to make that occur? What's going on to make that occur is negative feedback. This is generally accomplished by something that we call a reflex arc. And a reflex arc is all of these components that are in your body that help to assess, report, and then change a variable so that it stays within the acceptable range. So you probably or maybe looked at a reflex arc when you were in anatomy, because they had a nervous system reflex arc, and when you had a sensory receptor and then an afferent pathway going to an effector. So let's look at it physiologically. This is an example of one, but I want to do an easier one before I look at that one. So So we're going to use a reflex arc. And first, we're going to talk about regulating your house temperature, and then we're going to take that metaphor and we're going to apply it to a real body. So you know what happens in your house. So let's say it's the middle of winter, and you are trying to keep your house at 74, okay? But you've got children, and because you've only told them 3,000 times not to leave the door open, they did leave the door open as soon as they left, right? And it's 19 degrees outside, and then your house temperature starts to drop, right? And as your house temperature starts to drop, you need to initiate this whole negative feedback system, but you need some components in order to do it. The first thing you need is something that can detect the change in the variable. That is the thermometer portion of your thermostat. Yes, the thermometer portion. The thermometer does not change anything, it just measures. Do you see? Okay. And the thermometer now says 71. Okay. Too cold, not in the acceptable range. So it now says 71, but I haven't fixed the problem. I haven't even told anybody about the problem. So the next thing you need to do is you need to report the problem. And you need to report the problem via what we call an afferent or input pathway. And you are going to report it to the brains of the operation. In this particular instance, the brains of the operation is the thermostat portion. The thermostat portion that you set at the ideal temperature, right? So it's got the ideal, and it can compare the measured variable to the ideal variable. And obviously it has set in there somewhere, but I don't know anything about HVAC, so I don't know. The acceptable range around there. Does anybody know that? What it's actually set to? I don't either. So whatever. 71 is generally going to be too far away, right? So at 71 degrees, it's set at 74. You report it to the part that knows what it should be and what's an acceptable range around it. And that is called the integration center, right? It's the brains of the operation, although it's not necessarily in the brain. Okay? And so the integration center this time says identifies it as cold. And then it assesses it, and it says, too damn cold. And now it says, do something about it. And so the do something about it goes via the next step, and the next step is the output or efferent pathway. And the output pathway is going to go to effectors that can effect the change that you want to occur. In our house example, efferent pathway will go to what can fix the problem. What can fix the problem here? The furnace or the heater, right? Because it's too cold, right? So I'm going to send it out to the furnace or the heater. Those are my effectors. And those effectors are going to do their job, and they are going to increase the temperature in the house. But this wouldn't do you a damn bit of good unless that new stimulus, the new number for the variable, was fed back into the system. Do you see? Mm-hmm. So now it goes up to 72, goes up to 73, 73.5. Feeding it back into the system. As soon as it gets in the acceptable range, what does the whole thing do? Shuts off. If it wasn't a reflex arc that went all the way around in a circle, every time you tried to fix a problem, you create the opposite problem. So do you see that the new variable has to feed back into the system? And do you see that if you were doing that effectively, what happens is whenever your body temperature goes down, you bring it back up, but you don't continue to go up, right? That makes sense? So that is your reflex arc, the components of a reflex arc. Now what I want to do is apply it to the human body and body temperature and start to put the parts together with the human body. So if we're doing body temperature regulation and we want to understand what reflex arc makes that happen with body temperature regulation, first thing is the sensory receptors. Here we're looking for temperature receptors. And we have temperature receptors a whole bunch of places in our body. But primarily the ones that we have are actually assessing the temperature of the extracellular fluid. Some blood plasma, some interstitial fluid, that's what they're doing. And they're all over the place. And they're all going to sort of gather this information. And they're going to send it generally via the nervous system, right? Generally via the nervous system up an input or afferent pathway to the part of the body that knows what your body temperature should be. What is that part? What is the part in your body that knows what your body temperature should be? Anybody know? It's the hypothalamus. Yeah, it's the hypothalamus. Interestingly, the hypothalamus, which anatomically was pretty damn unimpressive, can we agree? It's like that? Really? That's that? It does everything. In physiology, if you don't know the answer, just guess hypothalamus. Hypothalamus, cellular respiration. I don't know, Melanie. One of those is probably going to be true. So, yes, it's hypothalamus. And the hypothalamus is basically the integration center for body temperature. The hypothalamus has stored your ideal body temperature, and it has set an acceptable range about that ideal, okay? And if my body temperature, for instance, was too low, I was outside getting really cold, then what's going to happen is it's going to say, okay, you're supposed to be at 98.6, that's what I wanted for you, you're down to 97, okay? At 97, what it's going to do is to say, too damn cold, just like the other one did. And now it's going to send via the efferent or output pathway, right, a stimulus to anything that can help with that situation, any effector that can help with that situation. So what effectors can you imagine could help with that low body temperature? What would you do if you were cold? Skeletal muscles, you would shiver. Goosebumps, hair follicles, you would do that. It would work better if you were a dog, but you still do it even though you're human. It's not particularly helpful. What about blood vessels? Yeah, you would vasoconstrict to the surface and keep the warm blood here, keeping your core more warm, right? And assuming that all of those things were effective, then what's going to happen is your body temperature will creep back up. And then you feed that new number, right? That new number back into the system, which will shut down all of the things. Does that make sense? So that is a reflex arc. And you can see how a reflex arc like that would result in this, right? It would result in trying to keep you relatively constant despite external stimuli, right? Okay, and you can also see, because you know that heating and cooling your home is expensive, you can see why this is going to eat up around 60% of the ATP. Just sort of trying to keep things relatively constant despite the fact that the external environment is not relatively constant. Okay, so that is a reflex arc. And we're going to see tons of examples of reflex arcs. The primary mechanism that you use for input and output pathways is the nervous system, and then sometimes the endocrine system. You do it with hormones and nervous system, and we'll talk about several examples of that. And then the last part, this part where, remember, you have to feed the new measurement of the stimulus back into the system, that little part of the reflex arc is sometimes called negative feedback inhibition. Because when you fix it, it should inhibit the cycle. Does that make sense to you? Right? The new fixed number should inhibit the cycle. So a little logic question for you. Some of these are functioning constantly, and some of them only function as needed. Does anybody have any idea which parts of these would probably be functioning constantly? Constant stimulus. You can't stop the stimulus. What else? Sensory receptors in the stimulus. And there's no reason to measure them if you're not going to integrate them. So even though I don't always respond to the stimuli, do you think that I only, for instance, take my own body temperature every 2 hours or so? Can you see any problems with doing that? Can you see any problems with doing that? Yeah, so what I'm doing is I'm sort of constantly doing the first part of this, and then I only do the second part of it when necessary. Why would I be constantly doing the first part of this? Because if I didn't take my body temperature but once every 2 hours, I could have let it go completely out of whack and not be able to fix it, right? So I'm sort of constantly assessing it. So interestingly, even when you don't need to fix your body temperature, it's expensive, right? Because you have to constantly assess it, right? Constantly assessing your body temperature, because it's really, really valuable. All of these things, really important to keep them in homeostatic normal, and you can't make it normal if you're not assessing it, right? So the first part of the system is always going on. Okay, so now there is feed-forward mechanism. Sounds like the opposite of feedback, but it's not. It's actually a helper for feedback. So I'm going to teach you about this one. A feed-forward mechanism, you would get the benefit of a feed-forward mechanism, which is also called anticipatory homeostasis. You would get the benefit of this if you were, for instance, very, very regular about your habits. So if you were a person who went to bed at 10:30 every night and got up at 6 a.m. every morning, then what would happen is your body would try to make that more efficient for you, because it would say, hey, it's 10 o'clock. I'm going to start dropping your metabolism because I know that that's what you're going to do, okay? Hey, it's 5:45. I'm going to start increasing your metabolism and start releasing the hormones and everything that are related to being awake, because that's what I think you need. And if it's right, 80-85% of the time, can you see how that might actually be effective, right? But if you're somebody like me who never goes to bed at the same time and never wakes up at the same time, then it would be completely inefficient to do that. So this is like predictive homeostasis. It's like, I think this is what you're going to need. And it's really helpful if you are very regular. Because, for instance, obviously it's working, because if you are one of those people who's super regular about your habits and you go on vacation, right, and you say, I'm going to sleep late every damn day on vacation, what happens? What time are you going to wake up? Same time you always wake up, right? Okay? But if you're me, right, and you never do it at the same time at all, and my alarm doesn't ring, what happens to me? I miss the whole freaking day is what happens if my alarm doesn't ring. So anticipatory homeostasis is based on regularity, and it doesn't work if you're not very regular. There are some examples where it will work in you, even if you're not one of those super regular people. If you walk into grandma's house, right, and you smell chocolate chip cookies baking, what's your anticipatory homeostasis response? I think I'm going to need to break down some carbohydrates pretty soon, so I'm going to start to salivate. And then she slaps your hand and tells you those are for Relief Society. You tell her she owes you some ATP, so you need a cookie. Or you can have the sexuality example. The sexuality example is, okay, so your incredibly hot wife puts on lingerie and starts parading around in front of you, and you have an anticipatory homeostasis response based on prior experience. And then she's like, nope, gotta go. She owes you a cookie too. No, I'm just kidding. She doesn't no cookies. So those are feed-forward mechanisms. Can you see that they are not the opposite of negative feedback? They are trying to increase the efficiency of negative feedback. Okay, so now let's talk about some types of negative feedback. So they all use these reflex arcs, but How do they use the reflex arcs? Short distance, long distance reflex arcs, what are you going to do? So there's two different types of negative feedback, and I'm going to tell you the names and the descriptions, and then you are going to match the names with the descriptions. One of these, if I have a problem in an organ or a tissue or a body system, I handle the problem within that body system. Okay, that's example A. Have a problem in a body system, handle it within the body system. The other one is if I have a problem in a body system, I call on the nervous system or the endocrine system to help me with the problem. That's situation B. And here are the names. One of them is called intrinsic regulation, and the other one is called extrinsic regulation. What is situation A? Intrinsic, yes, intrinsic regulation. And situation B is extrinsic regulation. That makes sense to you guys? Okay, now let me give you a metaphor for that. So, if somebody breaks into my house, I call the cops. If somebody breaks into Tony Soprano's house, does he call the cops? No, he handles that crap in-house, right? That's intrinsic. I'm going extrinsic. Does that make sense? Okay, so let me give you an example of intrinsic and extrinsic, a few examples of each. So with extrinsic regulation, it's generally long-distance communication because you've got a body system that's calling on the nervous system or the endocrine system. The nervous system and the endocrine system are the two big helpers with homeostasis. So the examples of extrinsic regulation. Okay, you were a child, so you know that we can live through this situation. It is Halloween, and I have siblings, and I got 6 full-size Butterfingers. And how many of them do I eat? All 6, because that's the best place to keep them when you have siblings, right? And so that is going to cause a massive disturbance of homeostasis via my digestive system, yes? And I'm not going to be able to poop them out fast enough to fix it with my digestive system. Do you agree? Okay. Massive disturbance in my digestive system is primarily going to be that my blood sugar level, my blood glucose level goes through the roof, right? But you're not going to die. This is a healthy person, not a diabetic child. So blood sugar goes through the roof. And what's going to happen is, can't take care of that with the digestive system quick enough to make it effective. So what's going to happen is I am going to call on the endocrine system. I'm going to send an input pathway to the pancreas. And then the pancreas is also the integration center. And the pancreas is going to say, oh my God, too damn high for blood glucose, and it's going to release insulin as a response. And insulin is going to go into the bloodstream and to almost all the cells in the body, and all of the cells are going to suck up the glucose from the extracellular fluid to the intracellular fluid, from the blood plasma to the intracellular fluid. And then what's going to happen to my blood plasma glucose level? What's going to happen? It's going to drop, right? It was way too high, and now it's going to be low, right? But that's not going to help anything unless, of course, you feed that number back into the system, right? You're going to feed that new blood glucose level back to the pancreas, and when it gets back into acceptable range, what is the pancreas going to do? Stop releasing insulin. Does that make sense? Okay, so a couple questions for you. First question. Can you see that this is an example of negative feedback? Can you see? It went high and you brought it back low. Second question. Can you see that this is an example of extrinsic regulation? Somebody explain to me why it's an example of extrinsic regulation. Go ahead. Yeah, and it's because you're using a different body system, right? Right. Digestive system disturbance handled with the endocrine system, right? Does that make sense? Okay, so I don't want you to get into the habit of thinking that this is negative feedback because I brought the variable down, so let's do the opposite example, okay? The opposite example would be fasting, which is going to severely disturb your blood glucose. I've heard you guys do that around here. I'm a Texas heathen, so we don't do any of that, okay? So fasting, which is a digestive system disturbance, and you can't or won't or aren't going to eat. You're not going to solve it with your digestive system for whatever reason, okay? Your doctor told you you had an appointment at 11. It's 12:30 and you're waiting in the waiting room, right? You're about to eat him. So the fasting, the digestive system problem, and I've got really low blood plasma glucose. Now that's problematic. And does your body just say, well, it was a good run, I guess my brain is going to starve from glucose? It does not say that. It says, okay, you're not going to eat. I'm going to solve this problem for you with the endocrine system. So what's going to happen is this low level of glucose is also going to be detected by the pancreas, still the integration center for blood glucose. This time the pancreas is going to say, too damn low. That is not good. And then the pancreas is certainly not going to release insulin, because that wouldn't help the problem, right? Right? Insulin would not help the problem. It's going to release a hormone that you may or may not have ever heard of. It's called glucagon. It's not that hard, because you need it when your glucose is gone. It's a good way to remember it. So glucagon is going to go into the bloodstream, and it's going to go to the site in which you stored glucose. Anybody know the primary site where you store glucose? Liver. Exactly. The liver stores glucose as glycogen. And it's going to say, liver, dump some of that glucose into the bloodstream, right? And it dumps it into the bloodstream. And then, of course, this whole thing does no good unless, of course, you feed it back into the system, right? This new level of glucose is now going to go back into the system. And as soon as it gets back up to acceptable levels, what will the pancreas do? Stop sending out glucagon. Exactly. Does that make sense? So the reason I gave you this one is because I want you to see that in order for it to be negative feedback, the variable certainly doesn't have to go down, right? In this one, the variable needed to go back up, right? So questions, same questions. Can you see that this is negative feedback? Yes. And can you see that it's extrinsic regulation, right? Digestive to endocrine. Now, I didn't give you any nervous system examples here. You can use the nervous system for extrinsic regulation as well. But the nervous system examples are really complex, so I'm going to wait for a little while. You'll see them all semester long. You're just not going to see them today. Okay? Everybody good with that? Okay, so now let's do intrinsic regulation. Intrinsic regulation is when you have a problem or disturbance of a variable and you handle it with the local cells or tissues that are having the problem. So it's also sometimes called autoregulation. Does that make sense? Or local regulation, because it's local. So it's generally short distance communication, whereas the other one's generally long distance communication. And so here's an example. Okay. Sometimes cells in one area need more oxygen because they're very active, and the rest of the body does not necessarily need more oxygen. So here's a good example of a situation in which cells in one area would need more oxygen and the rest of the body's really not doing very much. What if I was ill yesterday or felt like crap and I took way too many acetaminophen? It's going to be really hard on my liver, right? Okay, acetaminophen is really hard on your liver. And so what's going to happen is I feel like crap, so the rest of me is just laying on the couch. But my liver is working its little liver butt off, right? And so it's going to need a whole bunch of oxygen for cellular respiration and ATP. You guys with me? Or you could do it the fun way. It would result in the same thing. You could do 6 shots of tequila last night. Still, you're going to be flat out on the couch doing nothing, except your liver is going to be working its little liver butt off, okay? So what's going to happen is I've got a local problem. I don't have a systemic problem. I just have a liver that needs more oxygen. And so what's going to happen is the liver is going to respond to low oxygen by releasing chemicals that cause local arterial dilation. Local arterial dilation. You with me? And what is local arterial dilation going to do to your oxygen content and why? It's going to get you more oxygen. How? I mean, I know you know. I just want you to verbalize it. More blood flow, more oxygen, right? And so now I'm going to get more oxygen into the tissue. And then as soon as I have an acceptable level of oxygen, what do you think those cells are going to do? Stop releasing the chemicals that cause vasodilation. Does that make sense? That's a little negative feedback example, is it not? So same questions I've been asking. Can you see that that's negative feedback? Oxygen level was low, you brought it back up. And then second example, can you see that this is intrinsic, not extrinsic? Yes? Okay, so that's the easy example of intrinsic regulation. Now we're going to do a complex one, and the reason we're going to do the complex one is because you have to do this particular thing all semester long. You might as well get it out of the way, right? So it's not going to be the last time you see it. This one is a little bit more complicated. So now we have to go back to our idea of metabolism and cellular respiration. And so you guys know that when a tissue is metabolizing, it's going to, of course, use up oxygen, yes, and generate carbon dioxide, yes? Okay. So as the tissue is more active, then the carbon dioxide concentration is going to go up. You good with that? Okay. So what's going to happen is as the carbon dioxide concentration starts going up, it is going to combine with this ubiquitous substance everywhere, right? Can't really avoid it. And then what's going to do is it's going to go through this chemical reaction. CO2 plus H2O forms H2CO3, which is carbonic acid. And the problem is going to be that carbonic acid does what acids do, which is drop H pluses, right? And then that's going to have an impact on your pH. Yes? Okay. So this is going to dissociate to H plus, which is the problem, and HCO3 minus, which is a weak buffer. Okay. So now I want you to see this relationship. That means that this is the relationship between carbon dioxide and H plus. As carbon dioxide goes up, what happens to H plus? As carbon dioxide goes up, what will happen to H plus concentration? It's going to go up, yeah? That makes sense? And then you need to do that little mental gymnastics that you have to do to figure out what that will do to pH. What will it do to pH? As H plus goes up, pH goes down. Yes? Okay. Everybody hates pH. It's not just you. Because you have to go, just flip it around. I don't know. Because it's the negative inverse log of the concentration of hydrogen ions. And everybody hates it, not just you. So here's the deal. How does this fit into our example? Because what's going to happen is as soon as pH starts to drop, it will be detected automatically by the respiratory system without using the nervous system, without using the endocrine system. And when you detect low pH in the respiratory system, it is going to automatically cause an increase in respiration, which will do what to your carbon dioxide concentration? Decrease your carbon dioxide concentration, and ultimately, what's that going to do to your pH? Increase your pH. Okay? Does that make sense? Okay, so, and as soon as you get your pH back in the range that's acceptable, then what will happen to your respiration rate? It's going to go back to normal. Does that make sense? Okay, so this one is a little harder to conceptualize, that it is negative feedback, but it is, right? Because your pH went too low and now you're bringing it back up, right? Why is it intrinsic? The reason it's intrinsic is because you don't need to call on anyone else. It's an automatic response of the respiratory system. It doesn't use the endocrine system, and it doesn't use the nervous system. It's constantly monitoring pH in the respiratory system, and it can cause an automatic increase in respiration. This is one of the primary mechanisms by which you regulate your pH in your body. Does that make sense? Okay, so this equation you're going to see all semester long. So you might as well sort of like just chew it up right now, right? Just get it out of the way. You can go through that thing. You probably did this. When somebody tries to teach you, for instance, cellular respiration, you're like, I am going to memorize that, but I am not going to learn it, and you cannot make me learn it. You can't. You're not going to do it, right? And then it just keeps coming back to torture you. Every single freaking class you take, this one's going to be like that all semester long. So just get it out of the way. Okay, so do you need practice on pH? If you do, again, interactive physiology has some good practice in which you can mess with it and watch what happens. You can do that if you want to. Okay, so now, what do most organs use? Intrinsic or extrinsic control? Yes, both. They're really going to try the cheapest, fastest one first. So if you had a disturbance, what would you try? Intrinsic. Intrinsic, which one do you think is faster, and which one do you think is more robust? Which one's faster? Intrinsic is faster, but it's kind of like weak sauce, right? It's only got so much it can do just locally. And then extrinsic is really robust because it can call on all kinds of things to help you. So what generally happens is that an organ, if possible, will try intrinsic regulation. And if that fails, it says, little help here, and then it tries extrinsic regulation. Does that make sense? So almost all organs have the capacity to do either or both. Not all, but almost all. Okay, so now we are going to switch gears and we are going to talk about positive feedback. And positive feedback is not homeostatic. Positive feedback means that you have to dump your normal, typical use of the word positive because it's not necessarily good and it's not necessarily up. What it is going to do is negative feedback opposed or negated the change, and positive feedback is going to add to it. So with our house example, what's going to happen with positive feedback is if in the middle of winter my kids leave the door open, the temperature stops dropping, instead of the heater coming on, the air conditioner is going to come on. It's going to add 2, whichever direction it was going, right? In the middle of summer, leave the door wide open, and temperature starts to increase, and the heater comes on, right? So it's going to add to whatever was going on. So you can see how it would be pretty damn impossible for that to be homeostatic, right? So what's going to happen is this. So let's talk about an example that you may have experienced. So let's say body temperature regulation. There are times when you lose control of your body temperature regulation, and they can go into a positive feedback cascade or a positive feedback cycle. So what can happen is you're going along, everything's just fine, your body temperature is going just fine, and then you get some kind of pathogen, and in your response to the pathogen, your body temperature starts to increase. But instead of, for instance, starting to sweat, what can sometimes happen? You start to shiver, right? Okay, and your body temperature goes up further. And for a little while, you might be in a positive feedback cascade. Most of us are healthy enough that we will catch that and pull it back into negative feedback eventually. But not all of us, and not in all circumstances. So, if you were a child, a 3-year-old, right, and your body temperature goes up to 103, 104, that's a big damn deal because their homeostatic control mechanisms are not as fine-tuned and not as robust as yours. So very often what happens is with a positive feedback, if you don't catch it, it can end in death or you need medical intervention. So it might be medication that you have to do right there to bring it back in, right? Or you might have to do that horrible thing that almost every parent has had to do, is dump them in an ice-cold bathtub because their temperature is too high. You don't want their little brains to boil. It's like one of the hardest things you ever have to do. But so the same thing with the elderly. When an elderly person's temperature goes up or down, it's a bigger deal than when yours does. Their homeostatic control mechanisms don't work as well, and they are not as likely to pull it back in as you are. If you are 25 and otherwise healthy and you get a freaking fever and it's 103, you'll probably be just fine if you just like sit there on the couch and just wait for it to pass. But your 3-year-old and your 85-year-old probably will not. But it doesn't have to always go up. So let's talk about a blood pressure situation. In a blood pressure situation, let's say my blood pressure starts to drop. I get into a car accident and go into shock. And I've got massive vasodilation, right? My blood pressure drops really dramatically. And instead of responding to put it back into homeostasis with negative feedback, what you would normally do if everything was working really well is your heart rate would increase and you would do vasoconstriction and bring your blood pressure back up. But if you are otherwise unhealthy, what can happen is you can respond with positive feedback and drop your heart rate and do more vasodilation. And then what's going to happen when your blood pressure continues to drop is you are going to die unless there is medical intervention at that point. So you can see why with humans, positive feedback is not homeostatic, right? Okay? As long as you're in the positive feedback part of it, you are not doing homeostasis. I'm not saying you're always going to die, but if you're in that part, you're not homeostatic. And it's always going to end in an event. And the event is either you got hold of it again, right? Or you had medical intervention, right? And you can see why very often positive feedbacks with humans is disease situation. Now, there are a few normal positive feedback situations. I'm not saying they're homeostatic, but I'm saying they're normal. So sometimes when you start something, you want to finish it very, very quickly. Yes? And you can see how if you started something, you wanted to finish it really quickly, positive feedback might actually help. So let's talk about blood clotting. So with blood clotting, what happens is you get a hole in a vessel, right? And then what you need to do is you need to plug that hole so that you're not losing any fluid. So here's how it occurs and see if you can hear the positive feedback in it. With blood clotting, what happens is the tear in the hole will attract platelets, and the platelets will release chemicals that start clotting and attract other platelets, which will release chemicals which will continue clotting and attract more platelets, and continue clotting and attract more platelets. So can you see what's happening? Is you are increasing the speed of clotting until what occurs? So the hole is plugged, right? Because clotting is not one of those things that you want to fart around with, right? When you want a blood clot, you've got a time increment that you want a blood clot in, right, before you die. Here's another example, birth. Once you go into labor, you do not want to stay in labor. That is not something you want to just dilly-dally around with. So here's how labor occurs when the baby gets to demand labor and not the doctor who's going on vacation, right? Here is what happens. A full-term baby will release oxytocin, and oxytocin will cause uterine contraction, and uterine contraction will cause the release of more oxytocin, which will cause stronger and more frequent uterine contraction, which will cause the release of more oxytocin. Do you see the ramping up? And the contractions get stronger, and they get more frequent, and they get stronger and more frequent, and it's going to end in an event. And the event, of course, is birth, right? And you can see how in that situation, positive feedback is normal. But when you're in that phase, you're certainly not having negative feedback for oxytocin, right? You're having positive feedback until you finish the event. And there's one other normal example. I can't teach it to you now, but I'm going to say it so that when I teach it to you later, we're going to rehook it to positive feedback. And that is generation of action potentials in neurons. Action potential is a great example of positive feedback, but it takes like an hour or maybe two to explain. So what's going to happen is I'll explain it later, and then I will remind you that it's positive feedback, and you will get to see why it's an example. Okay, so with positive feedback, what happens is you're going along just fine, and then you lose control, and it goes one direction or the other. It doesn't really matter which one direction it goes. Okay, so that's positive feedback. Now, let's go back and make sure that we can put all this together. So homeostasis is, of course, the maintenance of a relatively constant internal environment. But what I mean by constant can be kind of counterintuitive. I don't mean constant. I don't mean that I'm always 98.6, right? It's relatively constant. And the way I think of it sounds like an oxymoron, but to me it sounds true. And that is your internal environment has predictable variability. It's going to vary, but it's going to vary in a predictable way, right? Over the course of a given day, I'm not going to go up to 106 degrees or down to 74 degrees, right? So it's got predictable variability, right? So your textbook sometimes refers to that as a steady state. So homeostasis is going to be the unifying theme of physiology, and it's the biggest predictor that you have. So if you ever wonder what's going to happen physiologically, always guess the homeostatic response. If your blood pressure goes up, what's going to happen? You try to bring it down. If your estrogen concentration goes down, you're going to try to bring it back up, right? That is your best predictor. And usually, almost always, it's going to be correct. So what are you monitoring and trying to homeostatically control most? It's mostly ECF. It's most of what you're doing. All of those examples that we were talking about, blood glucose concentration and body temperature and pH, all of that was extracellular fluid that we were monitoring and trying to regulate. So the things that we are regulating are pretty much any variable that you can imagine is regulated by these mechanisms. And then also pressure and temperature and those kinds of things are also regulated. Fascinatingly, your body is regulating your own, it's measuring and regulating your own body temperature right now, your own blood pressure right now, your own pH right now, your oxygen content. All of that is going on behind the scenes. It is phenomenal that it goes on without my intellect because I know that I couldn't do anything if I had to do any of that, right? I can hardly walk and chew gum at the same time. So thanks, body. Okay, so let's assess a couple of things and make sure you've got the idea. So we know that constant doesn't mean constant. It means predictable variability. Your textbook describes that as a steady state, right? A steady, wiggly state that's going on for any variable. And so now the thing to ask you is, what is it actually trying to do? Is it actually trying to maintain my body temperature at 98.6, a set point? Or is it trying to maintain my body temperature in an acceptable range? Acceptable range, so we kind of understand that. There is a set point, but obviously that's not the only thing I will accept, right? Otherwise, every time I got slightly higher than 98.6, I would start to sweat, and slightly lower, I would start to shiver. But that's not what happens, right? There is a set point in there. There has to be, otherwise how could you describe the range? But interestingly, we don't speak hypothalamus or pancreas, so we will never know what the set point is. Does that make sense to you? We can't get it to spit out the set point. So what's the best idea we could get of the set point? Whole bunch of data over time, and then average it, and then you get the mean body temperature, and that's your best estimation of the set point. Does that make sense to you? Okay. So you take a mean over time, and that's why you have the 98.6 stuck in your head. But that's not my mean. That's the time averaged mean for a whole bunch of people over a long period of time. And so in a minute, we're going to talk about variability between individuals. Okay, so the mean and the range. And so you already know that the body maintains variables not at a true set point, but in a steady state around the range, right? And then, so here's the deal. A lot of times we have a couple of numbers memorized. We have 98.6 for body temperature. We have 120/80 for blood pressure. You may have blood glucose memorized. But if you're going into a clinical situation, most of you guys are going to, what do you think is going to be more useful? Set point, mean, or range? Yeah, you're going to need to memorize 2 numbers for everything, just so you know. Because you don't want to be the one that runs to the doctor, right, when the person is not at 98.6, they're at 100.3. You're like, oh, doctor, 100.3. No, you will lose your damn job is what's going to happen with you. In addition, you also don't want to be the person that runs to the doctor when somebody's got a blood pressure of 200/160, unless you ask them whether they just ran up the stairs, right? Your blood pressure is going to go up and it's going to come back down, right? So you need to know the range, ultimately. I don't have you memorize a lot of range. A lot of them are in the flaps of your textbook, but you will eventually have to memorize the ranges that are relevant to whatever your specialty is. And generally, blood pressure, of course, you're going to have to do. So range is more useful. Now let's talk about variability. So what does 98.6 have to do with anything? That's the time averaged mean of a whole bunch of people. It is not necessarily my mean or my set point. If you have lived with another human, you know that body temperature is variable between people, right? You know the thermostat wars that happen with your family, right? One of the leading causes of divorce, thermostat, sex, money, those 3, right? Because you think they're a freak of nature, right? I always want it like slightly colder than it is. And my husband's like, did you change the thermostat? Like, kids must have did it. They're like, they don't know the code. Oh, I can figure it out. They're smart children. Okay, so variability in me over time is absolutely normal. Variability between individuals is absolutely normal. But again, the variability is predictable. Like my body temperature is not an average of 98.6 and yours 103, right? It's still going to be pretty close. But my mean over time would be relatively constant. My mean body temperature over time would be relatively constant. And the more data you get, the better idea and the better mean that you get. So you know this, but let's talk about it anyway. Which is more likely to indicate disease? Variability from set point, mean, or range? Range. Only if what? If you vary from the range, yeah, if you vary from the range for a while, right? So I don't have hypertension if my blood pressure is 200/160 when I'm working out. It's just going to do that. I have hypertension if my blood pressure is 200/160 at rest over a long period of time, right? Then I have hypertension. Does that make sense? Okay, so you already knew that. Okay, so now let's talk about bodily processes related to homeostasis. So you guys are doing this in lab this week. You are starting to measure some of your own variables. Circadian rhythms. Circadian rhythms are variables that tend to vary over a 24, 25-hour cycle. Circadian means around, like going around the clock. Circa means around. So some of your variables vary over 24, 25 hours. Some of them vary over much longer time periods. Like for instance, your cortisol concentration, which is a metabolic hormone, varies over 24, 25-hour cycle, but estrogen doesn't. Estrogen varies over like a 28-day cycle, right? But the ones that vary over a 24, 25-hour cycle we call circadian. So that means that you must have a pacemaker or a time setter in you. That's the hypothalamus too. The hypothalamus takes input from light and temperature primarily to tell you when it's daytime versus nighttime. Of course, daytime tends to be more light and higher temperature, and nighttime tends to be the opposite. And the hypothalamus uses that to set your cycle, right? And then the hypothalamus controls, among other things, your pineal gland and tells it when to release melatonin, yeah? And you release melatonin when it's time to go to sleep. And then you go to sleep, and then the next day, of course, your melatonin's going to drop, and then you increase it again. So that's part of how you keep time. So hypothalamus and these circadian rhythms are helping to regulate your sleep-wake cycles and your body temperature, and some hormone concentrations vary over a 24, 25-hour cycle. And then, of course, this is where you can get into your anticipatory homeostasis. If you are really regular with your circadian rhythms, then your body will have a very good idea what is going to be needed next. And then you can start to do those feed-forward mechanisms or anticipatory homeostasis. Okay, so here's the next thing, and this is something that's really interesting and we're going to see all semester long. Okay, what if I fail to regulate a variable? And I fail in the same way over a very long period of time. Not days, not weeks, but months, maybe years. Okay, so let's use this example. I've got an adult male who has low levels of testosterone. Testosterone is not just related to sex drive. In males, not so much in females because estrogen does a lot of it in us, it's also related to metabolism, it's also related to drive, it's also related to mental acuity, it's related to a whole bunch of different things. It's related to appetite, both sexual and other appetite. So we got a male who's got chronically low levels of testosterone. And the body has tried its damnedest to get that testosterone level up. It continues to send a signal to the testes that says, we need more testosterone. But for whatever reason, the testes can't make more testosterone or don't make more testosterone. So I've got this sort of chronic level of testosterone that's too low. And eventually the body goes, okay, really, you can't make any more testosterone. Let's switch to plan B. And plan B is if you can't regulate the variable, regulate the response to the variable. And the response to the variable tends to have to do with receptors. So, I have chronically low levels of testosterone, but I want a relatively normal cell response to testosterone. I can do one of two things. You guys choose. Either increase the number and sensitivity of the testosterone receptors or decrease the number and sensitivity of the testosterone receptors. What do you think? Increase. Yeah? Does that make sense? So if you have to, this is definitely not plan A, right? But plan B is if you don't have normal levels, how about I increase the response to the levels that you have? Got it? Okay. That is called upregulation, right? And it happens over a long time when you failed to regulate the variable that was too low. Does that make sense to you? Okay. And that is like going at homeostasis with the effect instead of the variable. Okay. It's not plan A. You don't do it quickly. Here's another example of this, the opposite example. And I know that you've heard of this one before, even if you haven't heard it described this way. And that is this. Okay, so you live in the South. You drink 6 Dr. Peppers a day. Okay, you started with it in your baby bottle. That's why everybody's fat down there. In addition, the food is amazing. So you drink 6 Dr. Peppers a day. And so what's going to happen is I'm going to have this chronic sort of blood sugar spike and then insulin spike and blood sugar spike and then insulin spike. But I'm not going to stop drinking Dr. Peppers because I'm just not, right? And so what's going to happen, not over days, not even over weeks, but over years, let's say a decade, right? That habit will eventually cause me to say, damn it, I've been trying to regulate your sugar and insulin the best that I can. And I have failed miserably in the same way consistently over a long period of time. And so now I'm going to go to plan B. And plan B is to mess with your insulin receptors. Okay? So now what I want is a relatively normal cell response despite the fact that I have excessive levels of sugar and excessive levels of insulin. So what am I going to do to my insulin receptors to try to get a relatively normal response despite the fact that I have excessive amounts? Am I going to increase the sensitivity and number of my insulin receptors or decrease the sensitivity and number of my insulin receptors? I'm going to decrease them. Right, exactly. Because they are just completely overwhelmed and I still want to have a normal response. So I need to down-regulate my insulin receptors. Okay? And that is type 2 diabetes. That's what that is right there. And so what's the problem with that? The problem with that is that anytime I'm not drinking 6 Dr. Peppers, like for instance when I'm sleeping, my blood sugar is going to crash because I can't respond to normal levels of insulin. And then I'm going to have booms of blood sugar and crashes and booms and crashes. So I've just made my cells insensitive to normal levels of insulin. I basically just worn them out. And now when they see insulin, they're like, hay in a haystack, seen insulin my whole life, bored by it, right? So they decrease their number and sensitivity. And then you get all of the same symptoms associated with type 1 diabetes, although typically also associated with that is obesity, right? Because most people do not drink 6 Dr. Peppers a day and also work out for an hour and a half. Just doesn't really go together very well, you know, just doesn't. They probably don't eat kale either, pretty sure, right? Okay, so that's upregulation and downregulation. We're going to see tons of examples of that all semester long. It's really important with hormone levels. Too much will downregulate, too little will upregulate. It's also really important with drug responses. This is part of how you can become acclimated to a drug, right? Continue to put it in. What's going to happen to the receptors? You're going to downregulate them, and then you don't respond to the same amount anymore, right? So we're going to see that all semester long. Okay, so next thing is what else is related to this that you have experienced in your life. The next one is adaptation or acclimatization. This is your capacity to change your set point for a variable when you are given constant inputs. And so I'll give you a couple of examples. One of them we experience with seasonal changes every single year. So with body temperature. My body temperature has this acceptable range, right? And let's say I'm set right there in the middle of it, my set point, my ideal set point. But just like in your house, in the winter, you can move it from 74 down to 70 because you're not made of money, right? And in the summer, you move it from 70 up to 74 because you're not made of money. And it's still within the acceptable range for you. It's just less expensive to maintain. Same thing happens with your body. So every single year when it gets hot, the first time it's like 85, you're like, oh my God, why do I live here? It's terrible. I'm sweating so hard. And it goes on for like 2 weeks, right? And then what happens is your body goes, really? You're going to stay in Utah in the summer for real? For real? And I'm like, yep, apparently, own a house. And it goes, okay. And it just moves your body temperature up a little bit. And then there's not as much variability between the external temperature and you. And you don't have to use your air conditioner so much, right? Does that make sense? In the winter, the same thing happens. When it gets cold, you hate it every single year. You're like, oh my God, it's so cold. I can't believe it. And then you're freezing, and you're freezing, and you're freezing, and then all of a sudden you're like, I'm fine, it's good. I can still go out and get the paper, right? I can do that. I can check the mail. So then what happens then is you move your body temperature down a little bit. You don't do it when you have a 1-day change in temperature, because can you see how inefficient that would be? What you need is a relatively constant difference that's pretty much the same over a long period of time. And then you can say, oh, this has really changed this external environment, then I'm going to change. But you don't change it with a single day. It's just not efficient to do that. And then the other one, a great example, is how many of you guys are not from Utah? Oh, there's a lot of us. Okay, so anybody move from sea level or close to high elevation and couldn't freaking walk up the stairs for like the first 2 weeks when you got here? So it's a very common experience. So I moved from Texas. It's pretty close to sea level. And the deal with being at sea level is there's tons of oxygen in the air. And that means that I don't need that many red blood cells to carry the oxygen around, right? So my acceptable level of red blood cells was set pretty low, right? And then you move here, and it's high elevation. And at higher elevations, there's way less oxygen available in the air. And so all of you who have lived here your whole life, your set level of red blood cells is much higher. So I moved here from Texas, and I moved to, well, there were a lot of problems. First of all, I didn't know what I was getting into. And I moved to Provo, where they don't like my kind. And no matter how many times I invited them over for cocktails, I couldn't make a damn friend to save my life. So in Texas, you swear to show you're real, and you invite people for cocktails to show you're friendly. And it just does not work here. I don't know why. But now I moved to the Avenue, so it's totally cool. I'm fine. I got my friends there. So, but I could not figure it out. So that was miserable. And then also, I'm like, oh my God, I'm never going to breathe again. So that went on for a couple of weeks. And then what happened is my body was like, are you going to stay, Melanie? For real, for real? You're going to stay? I'm like, apparently I got a job. I'm going to stay. And so then what happened is my body's like, okay, well, you're going to need some more red blood cells. And so what happened is it moved the set point up for the acceptable number of red blood cells. And my bone marrow starts working, and I get more red blood cells, and then all of a sudden I can breathe and walk. And it's great, except I still lived in Provo. So there was that. But one thing at a time, right? We handled that later. So, okay, so those are adaptation or acclimatization. It's your capacity to change your set points with constant input of data that varies from your set points. Go ahead.
Regarding temperature, if our normal body temp is closer to 98, why are we not super comfortable when it's 98 outside? Why are we not like, ooh, this is our spot?
because you are constantly generating heat. So your normal body temperature is 98, but you work your butt off to keep it at 98 because you're generating a lot of heat. And so you're cooling yourself down constantly. And so you're cooling yourself down to close to the ambient temperature. When the ambient temperature is hotter, then you don't have the ambient temperature helping you with the cool down. So you are going to be really, really hot. Because generally I use the ambient temperature to cool down my surface and then take that cool blood back and circulate to the middle. So now you've got double hot. That's what's going on.
Is that why like 75, 70 seems to be comfortable because you're...
Because it's got this good ambient temperature that helps you maintain your body temperature best. And so that's kind of what you need for maintaining. They say that that for sleeping, the ideal is slightly lower. Your sleeping temperature is usually closer to 70 because your body temperature actually needs to be slightly lower when you're sleeping. And your awake temperature is somewhere between 73 and 76. I like it colder than that. I would prefer it's freezing and I put on blankets at all times, you know. And I drive a hybrid car, so don't judge me. I'm still going to run my damn air conditioner. Okay. I'm going to do it. Okay. So aging. What does aging have to do with this story? Aging is there is a progressive deterioration of your homeostatic control mechanisms. They are wearing out just like every other machine, right? And so what happens is as they wear out, you go out of range, but you stay out slightly longer than you used to. And when you stay out of range slightly longer, you have the capacity to cause a little bit of damage, right? So you go out of range, you cause teeny bits of damage, you go out of range, you cause teeny bits of damage, you go out of range, you cause teeny bits of damage. And this sort of progressive deterioration is going to sort of wear out your parts and continue to wear out your homeostatic control mechanisms. And that's what aging has to do with the story. You've got this period of your life where everything is homeostatically controlled in this sort of ideal situation, right? And then basically in your 30s, it just starts to fail you. And then it's going to fail you progressively from then on. Now, it's not a death sentence. If your cardiovascular system is in amazing shape, you will put that off for later and later. But I don't know anyone who's lived eternally. So you're going to fail eventually. You're just going to. Okay, so that is aging. So the last thing today, and I'm briefly going to start you guys on this. I'm not going to go into it in tons of detail, is now we need to talk about how you can accomplish this. Like, so there's communication. There's like the input pathway and the output pathway. But what are you actually using for the pathway? So you've got 100 trillion cells with sort of one overriding goal, which is to maintain homeostasis for the entire body. But the cells are specialized because my liver cell does not do the same as my skin cell, as my heart cell. So they're all specialized. But if my liver cell malfunctions, it will affect my heart and my skin cell, right? Because I won't decrease the toxin. If my skin cell malfunctions, that, for instance, it doesn't make melanin, the melanoma or damage from UV radiation will affect my entire body. Same with my heart. So what happens is if I've got one overriding goal and I've got specialized cells, that means I need a lot of communication. Do you see the necessity for communication? If I do this, you do that, and we all have the same overriding goal, then we're going to have to communicate with each other. And so the next question is simply, what is it that allows cells to communicate with one another? And the answer is chemicals. So if I'm a liver cell and she's a heart cell, how do I communicate with her? I communicate with her via the release of chemicals. I release chemicals called intercellular. What does that mean? Not inside the cell. Between the cells. Intercellular chemical messengers. So I'm going to release a chemical messenger, but she can only hear it and respond to it if she has a what? Receptor. Yeah. If, for instance, she's a heart cell and she's got receptors for what I'm releasing, if she is a testes cell and she doesn't have receptors, she will never even know that I said it, right? Does that make sense? So you've got this specialization that calls for communication, and communication occurs via these chemical messengers that interact with a target cell that has a receptor for the chemical messenger. Does that make sense? And now let's talk about the categories of chemical messengers. And the categories of chemical messengers are mostly things you've heard of before. How do we communicate between cell A and cell B? One big category of chemical messengers is going to be neurotransmitters. We're going to talk a lot about neurotransmitters. Next big category is hormones, right? Communicate between cell A and cell B. And the third category seems to be less familiar to students, but it's actually the most ubiquitous one. It's the one that we have the most of. And these are called paracrine and autocrine agents, okay? So those are going to communicate between cells. Some of them are long distance between cells, and some of them are short distance between cells. But these are all chemical messengers. So we are going to, of course, this semester spend a lot of time talking about the communication between cells. And then we're going to be talking about transport and that kind of stuff. So next time what we're going to do is we're going to start setting up how do you classify things as hormones and neurotransmitters and paracrine agents and some examples of each. And then after that we'll move on to cellular metabolism. But your big goal before next Wednesday when I see you, because you've got a long weekend, is clickers. I need for you to have your clicker on Wednesday, because when we meet on Wednesday we are going to register them in class, and I'm going to give you a mock quiz so that you can get used to the technology. And then the time after that, which will be the Monday after that, we're going to have our first graded quiz with the clickers. Okay? So make every effort to get them, please. See ya.